Today we want to talk about cartilage and bone. Cartilage and bone are the supporting connective tissue of the body. We want to talk about the various forms of supporting connective tissue with fibers and extracellular matrix in the supporting connective tissue, and talk about the types of growth of bone, and compare compact and spongy bone. Thank you. Cartilage, bone, and osteogenesis. We want to look at the histologic identification and function characteristic of different types of supporting connective tissue, cartilage and bone. We want to recognize the structure and the characteristics of the function of the cells, fibers, and ground substance. Describe the cellular mechanisms for intramembranous and endochondral bone development, and compare and contrast the structure and function of compact and spongy bone. Now, the functions of cartilage is a flexible support. It returns to the original shape. You can fold up your ear and it pops right back because it has elastic cartilage in it. The nose and respiratory system. So it's flexible support, not rigid but flexible. Also, it slides across each other easily while bearing weight, like in joints or the articular surfaces. It's a cushion somewhat. It's somewhat compressible, so it cushions the weight on the bones by having cartilage at the end of the bones. There's no nerves in cartilage, so there's no pain whenever you compress the cartilage. Now, connective tissue are of various types, and cartilage and bones is a supporting connective tissue. Before we talked about the connective tissue proper, and today we're talking about cartilage and bone. The cells of cartilage and bone come where other connective tissue cells come from, and that is from undifferentiated mesenchymal cells in the connective tissue. And of the bones and cartilage cells that we'll be talking about today, we have the chondroblasts, which give rise to chondrocytes. We have osteoblasts, which give rise to osteocytes. And another main player we have is the osteoclast, which is derived from the monocyte because it is a phagocytic cell that allows remodeling of bone. So in the general organization of cartilage and bone, we have the perichondrium. Here we see the perichondrium, near the chondrocytes, and it's dense, irregular connective tissue characteristic of a capsule. The perichondrium forms the connection with other tissues of the body. Also, it harbors a vascular supply. It's the source of nutrients that diffuse into the chondrocytes, which are located in the cartilage material. Now, the matrix is you have type 2 collagen. Type 2 collagen lacks the obvious periodicity. It does have periodicity, but not as elaborate as that of type 1 collagen that we see in bone. So we see the type 2 collagen fibers, and then you have a hyaluronic acid core with proteoglycans, and some of those are sulfonated. So you have sulfonated proteoglycans, congruent sulfate, keratin sulfate, and that's what gives cartilage its bluish basic staining that it has in there. And here you can see where the congruent sulfate, these sulfates are added to the protein core, which add to the hyaluronic acid, which is a ground substance that then connects to the type 2 collagen fibers. As I mentioned, it allows diffusion to occur. This matrix allows diffusion to occur, and also cartilage is avascular. As a consequence, it gets no nutrients from blood vessels. It gets its nutrients from the perichondrium and by diffusion of nutrients through the ground substance. Now, chondroblasts give rise to chondrocytes whenever they get trapped in the extracellular matrix. And the extracellular matrix is produced by amino acids coming to the cell and protein synthesis. Ultimately, proteins are being produced up through there. Sulfate and sugars are added in the Golgi apparatus, and then ultimately the proteoglycans are produced, and the type 2 collagen likewise is produced. So these cells, the matrix that trap the osteoblasts into their lacunae or space that they are in, and that at that time they become chondrocytes. Now there's 3 different types of cartilage. There's hyaline cartilage, which is kind of glassy, what you see at the end of a chicken bone, and that has a perichondrium around it. Here's a perichondrium here and here. There's also elastic tissue. It has elastic fibers, very similar to hyaline cartilage, but the groups of cells are fewer in these, and it does also have a perichondrium around it. Fibrocartilage is different in that you have individual chondrocytes that are embedded in connective tissue, like in a tendon or a ligament attaches to a bone. There is no perichondrium in the fibrocartilage because they're already in a connective tissue matrix themselves. So in terms of development of cartilage grows by 2 mechanisms. One is interstitial growth, that is mitotic division of pre-existing chondroblast, and that's what we have here. In fact, you have these isogenic groups. You could have multiple ones, all come from the individual cell by mitosis. The other type of growth is differentiation of a new chondroblast from the perichondrium. So you have these precursor cells in the perichondrium that we see here and here, and they differentiate into chondroblasts, which gets trapped in their lacunae and become chondrocytes. So if we look at the trachea, we can see the cartilage ring on the outside that actually holds the opening of the trachea. It forms a ring in that it does not go all the way around and make a complete circle. And so we have a series of these rings. We see one ring ending here and another ring beginning there. So here is the perichondrium. These are the chondrocytes located in through here. We see the Osteogenesis group, all those each in their own lacunae. And we have a territorial matrix, that is the matrix right around individual cells, and that tend to be a little more blue, and the interterritorial matrix, which tends to be a little more red, as we see here. The staining varies within the matrix related to the composition. Immediately surrounding those, the extracellular matrix is rich in glycosaminoglycans, more sulfonated, and so it's bluer. So territorial, a little more blue than the interterritorial, which have been diluted out a little. Remember, the proteoglycans with the sulfur is produced by these chondroblasts. The epiglottis gives an example of elastic cartilage. Elastic cartilage is here. We can see the cartilage from here to here, and it does have a perichondrium around it. But elastic cartilage has obvious elastic fibers, and you can see these fibers running through here, which allows it to return to the original shape. Like you can bend your ear, it pops right back. But individual cells are still in their lacunae, and you have fewer isogenic individuals in elastic cartilage versus the hyaline cartilage. And here we can see elastic cartilage again in slide 16 with a special stain, and we can see the elastic fibers inside there. This would be a chondrocyte in its own lacunae. This is the perichondrium located here and here. Now fibrocartilage is a little bit different, and it's intermediate between the dense irregular connective tissue and hyaline cartilage. And so you can see individual cartilage cells in dense regular connective tissue. There's no perichondrium because the individual cells are already embedded in a type 1 collagen matrix. And here we can see there's an intervertebral disc. Also the attachment of ligaments is what we see. So in a tendon or a ligament we see lots of type 1 collagen, and then we see the chondrocytes located intermittently in there, and around each one of those you would have type 2 collagen because that's what cartilage produces as opposed to type 1. So it's type 2 surrounded by dense amount of type 1 collagen. And in slide 17, the vertebrae, we can see There's some hyaline cartilage here. There's the chondrocyte located in this lacunae, and this red here is the connective tissue, compared to this red here, which is this is bone itself. So we have fibrocartilage, dense irregular connective tissue that the fibrocartilage is associated with, hyaline cartilage here, and this is bone. Another example of fibrocartilage here we can see in the fetal elbow, right in through there. So attaching the tendon or ligament to there, and we can see the fibrocartilage with individual cells, and then this dense regular connective tissue that is embedded in. Regular because it pulls all in one direction. Again, we can see the individual chondrocytes, dense regular connective tissue there. If we compare the fibrocartilage to fibroblasts in the tendon, there's a regular fibroblast that we can see. There's a little tendon that's coming in through there. And we compare that to fibrocartilage, you can see that these are uniform. The cells here, this is a young one, and so it hasn't have a lot of extracellular matrix yet like we see here, but then you can see the different cells, the chondrocytes in the fibrocartilage. So in summary, there's three types of cartilage. You got the hyaline cartilage, glassy type. You got the elastic cartilage that returns to its original shape, and the fibrocartilage, which allows dense, regular connective tissue to attach to bone. And that brings us to the cells, the chondroblasts, which secrete a matrix and become chondrocytes, and they could divide by interstitial growth as well as differentiation. Next comes bone, and one of the main players of bone is osteoclast. Osteoclast is derived from the monocyte, again from the connective tissue in the bone marrow. The other two cells are the osteoblasts, which secretes a matrix and secretes a matrix around itself, making a lacunae that is in as osteocytes. So these guys give rise to these guys. Now the function of bone is skeletal support of land animals, and you can see these guys in this heavy elephant is brought about by the fact that you've got bone. This is compact bone, this is cancerous bone or spongy bone on the inside. Also, it protects enclosures. It protects your chest with your ribs. It also protects your brain with the skull itself. Protects your brain. And the long bones is a source of hematopoiesis. So, before you can get blood cell formation, is protected in the spaces of the cancerous bone or the spongy bone. So the function of bone also is in regulation of calcium. The parathyroid hormone causes bone reabsorption. Calcitonin from the thyroid prevents reabsorption to occur. So bone reabsorption or prevention of reabsorption is the two things that happens. When bone is reabsorbed, you increase your calcium level. The hormones, these hormones are involved in tight regulation, as 1/4 of free calcium in the blood is exchanged every minute, and that's due to the breakdown and deposit of calcium in bone. Also, bone is important in hematopoiesis, which provides the sites for hematopoiesis to occur. There are certain cells in bone. You have the osteoblasts. These are the ones that lay down the new bone. And then you have expansion of bone by appositional growth. That is, that these guys produce bone around itself, and then as they grow on the edges, is appositional growth. So on the edge they grow. Cell is the osteocyte. So once the osteoblast is secreted its matrix, it gets locked in this matrix, and when it's trapped, it becomes the osteocyte. Osteoblasts give rise to osteocytes. And here we can see the osteoblast laying down the matrix. This is a type 1 collagen that's characteristic of bone, and here we can see where it's mineralized. Osteoblast trapped in the matrix. Osteoclast is a multinucleated phagocytic cell. It comes from monocytes and derived from the bone marrow. Remember, again, it's from the same cells, mesenchymal cells that are located in the bone marrow, come to the monocyte, and then it makes several of these makes osteoclasts, which eat up bone. If we look at the vertebrae, we can see fibrous periosteum. So we got the connective tissue core, dense irregular connective tissue that surrounds around it. There's cancerous bone that we see here, a spicule of bone, and here we can see osteoblasts lining the spicule of bones. This is primary spongiosum, and it still has a cartilage core. So the bone is replacing the cartilage, and here we see an osteocyte that's located in its lacunae or space. In developing bone, we have two different types. A typical type is endochondral ossification, where you form cartilage. In the embryo, we have a cartilage skeleton, and then that is ultimately replaced by bone. And in fact, the growth of long bone is dependent upon the cartilage growth at the ends of the bones, and the cartilage is then replaced to expand the length of the bone. Because there's no interstitial growth in bone, it only has appositional growth or differentiation from the cartilage itself. That is, it's replaced the cartilage bone. It doesn't differentiate from cartilage, but it replaces the cartilage core. Another type is intermembranous. That is, bone is formed directly from the connective tissue cells without going through cartilage. So intermembranous ossifications, direct mineralization of the matrix secreted by the osteoblast without the cartilage core. So you have mesenchymal cells giving rise to osteoblasts, which then produce extracellular matrix. It gets trapped osteocytes. The osteoclasts move in to remodel it, to digest parts of it, to remodel it, related to making calcium free for the blood. And here we can see in the skull is one of those examples where you have intermembranous ossification, flat bones of the skull, where you have differentiation of fibroblasts directly to osteoblasts. And in our slide, we can see in 36, we can see these osteoclasts here, which would be eating up bone, and these osteoblasts laying down new bone osteocytes in the spicule of these bone. Histogenesis of bone, the endochondral ossification. You have deposition of bone matrix on a pre-existing cartilage matrix is what we have. That's the characteristic of long bone formation. And here we can see the cartilage that will be replaced by bone. And you have a zonal proliferation to occur. What we don't see is a reserve up there, but we have zonal proliferation. Hypertrophy, where you increase in size of the cells. And then you have deposit of calcium in the cartilage to occur. That causes the cells to die because the calcium prevents nutrients from being diffused through, and the consequence, osteoclasts move in to get rid of these dead cells, and they're replaced by bone. Osteoblasts move in and then produce osteocytes. And here we can see it in our specimen of long bone 20, slide 20. We see the reserve cartilage. This is bone here, so this is a reserve source of the cells. We see zonal proliferation, where they increase in number, zonal hypertrophy, where they increase in size of cells, so increase the size. And then we get calcification to occur, and then the zone of ossification, where cartilage is replaced by bone. Osteoblast lay down new bone, and we see new bone being developed there. Now, the extracellular matrix of bone, the osteoid, the stuff that's secreted by bone, the extracellular matrix, is of type 1 collagen, and it's a complex mixture of minerals, material, to increase the affinity and serve as nucleation site for precipitation of calcium phosphate or hydroxyapatite. That's what we're seeing there. So this calcium is being distributed throughout the body by the blood, and it could have deposits various places, but most of the places of the body you have inhibitors for the nucleation of calcium deposit, the precipitation of it to occur. But in bone, The osteoid is especially adapted so that it will stimulate the hydroxyapatite formation to occur. And the cells are the osteoblasts are basically polarized in that they give rise to development of bone in the appositional approach. They add it to the outside. So as they produce this osteoid or extracellular matrix material, type 1 collagen, hyaluronic acid, and those materials, they become trapped. And when they become trapped in their lacunae, they become osteocyte at that point in time. But it's the calcium phosphate allows the hydroxyapatite crystals that allows the firmness of bone to be there. So extracellular matrix is not only the type 1 collagen and hyaluronic acid material, also it has calcium deposit. So you get calcium deposit, type 1 collagen as well, and as a consequence it becomes nutrients cannot diffuse through. So in order for cells, osteocytes, to be able to get nutrients, they maintain connections to adjacent cells, and then the extracellular matrix is laid down around it, and the type 1 collagen as well as calcium deposits. But there's a little hole. The lacunae or space continues on so that they keep connections to the adjacent, and the deposit is deposited around them, so the lacunae extends down from one cell to another cell, and we call these canaliculi. And that's the source of nutrients, is that one cell gets nutrients from another cell. It's passed through these gap junctions. There's a gap junction between adjacent cells, and here you can see these canaliculi which projecting from one cell to another one. So nutrients comes in through the Haversian canal, and then from there ultimately it goes from one cell to another cell. Nutrients are passed through gap junctions that occurs. So we have compact bone, which is on the outside, large bones, and then you have spongy bone. Spongy bone have spicules projecting up there with spaces in between, and the spaces is where you have the bone marrow occurs. Compact bone, you can see where you have the Haversian systems. These complete systems, a series of systems that go as the bone is developed. Osteoblasts are producing osteocytes by appositional growth, and as they grow they make these lamellae. These are circumferential lamellae on the outside, and that's part of the periosteum. So cells differentiate from the periosteum to give rise to osteoblasts, which then gets trapped as osteocytes, and that produces the outer interferential lamellae. There's no interstitial growth to occur, so bone can't expand because of the calcium deposits that are in through there. It can't expand like cartilage can, and so it can only grow by appositional growth. So it grows here by appositional growth, and it grows on the inside, and we call that inosteum. So what's on the inside of bone, covering bone, is the inosteum, and again you get deposition of bone in the appositional approach. We also, in these Haversian systems, you have a similar type thing. You have a hole, and then cells are added by appositional growth, one layer at a time. These are older cells in those cells, and this is also called the inosteum. The inosteum is inside the Haversian system, developing the Haversian system, laying down one layer, another layer, another layer. It lays down the layers on the inside as opposed to the outside. So it's just the opposite of a tree, laying down these different layers. And here you can see these are Haversian systems. Another word is osteon. And here's a whole system itself. There's another one right there. Haversian canal, the canaliculi, the osteocytes located in through there. And then the Volkmann's canal brings nutrients from the inosteum and the periosteum, as you see in through there, and it goes to the Haversian system, and that's the source of to get the nutrients. So nutrients come in from the periosteum, through the Volkmann's canal, through the Haversian canal, and then pass by gap junctions between adjacent cells to feed these cells inside. And in slide 19, we can see a compact bone. There's a Haversian canal, Volkmann's canal going through there, Haversian canal in the center that we see, circumferential lamellae. These are older than those, the younger ones are in the center. And you see the lacunae where the cells have been trapped, and then the canaliculi. So interstitial lamellae is where a Haversian canal, it's where a Haversian system was before, and is partly digested away now. So connecting adjacent osteons or Haversian systems, you have the Volkmann's canals. They provide communication, nutrients, blood for the Haversian canal, which ultimately feeds the Haversian system. Now, in terms of clinical correlations, we want to describe the cells that may be involved in the imbalance of calcium and define a couple words is what we want to do. Now, remember that calcium regulation is done by a couple hormones. The parathyroid hormone stimulates osteoclast production. So you produce more osteoclasts. So the parathyroid hormone comes from the parathyroid gland, chief cells therein, and they produce that, and that produces more osteoclasts. Osteoclast breaks down bone. So if you have more osteoclasts, you're going to have higher calcium levels. Calcitonin from the parafollicular cells of the thyroid, they remove the osteoclast rough borders. So here we see an osteoclast, multinucleated thing. It has a rough border here, and that creates a microenvironment with acid to break down the bone to dissolve the calcium. And if the rough border has been removed, then it reduces the effect of the osteoclast. So one makes more osteoclasts, and another one interferes with the function of the osteoclasts, the two hormones that regulate calcium. Now, osteoporosis is in balance between the two. You got more breakdown than build up, and it leads to calcium loss of bones and reduced minerals. In contrast, there is other defects where you have a defect in calcification of recently formed bone or partial decalcification of already calcified matrix. So bones protect the brain. We need to keep our bones healthy. Thank you.